In this video, we will discuss about this off-the-shelf allogenic NK cells for immunotherapy. So main problem with this off-the-shelf NK cells is the immunogenic response that it gets from the host immune cells. So the researchers tried to modify these NK cells or the allogenic NK cells so that it will have minimum immunogenic response when they are used for immunotherapies in the patient. So for that, the reasons for the immunogenic response is the expression of this HLA, that is leukocytes, human leukocytes antigens, which are being recognized by the T cells or the other immune cells, and then it will generate immunogenic response. So if this HLA can be suppressed, then it will have the less immunogenic response by the host immune system. And one more thing about this HLA is that there is one more surprising thing that is observed about this HLA is that the HLA ABC, the decrease in the HLA ABC will cause decrease in the immunogenic response, but there is one more HLA, it is HLA E, and decrease in HLA E will leads to the decrease in signaling pathways NK-GD2, and that will leads to these NK cells more prone towards killing by these immune systems. So we have to suppress this HLA ABC. Idea is to suppress the HLA ABC, but at the same time keeps minimum effect on this HLA E. So for that, they screen. Initially what they did is they screened different guide RNAs that will give a good results in terms of the reduced expression of this HLA ABC, and they are able to find out 4 different candidates that is mentioned in this panel A, and these things they have quantified in the panel B also. And in that case, the shRNA 13 and 14 is targeting BMP2, whereas guide RNAs 1 and 2, shRNAs 1 and 2 is targeting this HLA ABC directly. In the results, they have seen that this guide RNA 1 and 3 shows a high potency towards reducing suppressions of this HLA ABC on these NK cells. And thereafter they checked this guide RNA's 1 response against another variant of this HLA, and they are able to see a decrease in this signals of this HLA A star. This is the samples they took from the patient, and now they also checked its effect on this HLA E, and they are able to see that the HLA E effects is also can be seen. And in order to check this HLA E effect, what they did is they took the jurkat cells and treat them treat them with this IFN gamma, and that will lead to the high expression of this HLA E. And when they use the guide RNAs, they check whether this suppressions of this HLA-Es occurs or not. So in that case, they see some kind of suppressions, but when they quantified it, they are able to see that this guide RNase 1 has a minimum effect, whereas the guide RNA 13 has the maximum effect. Almost it will remove the HLA expressions on the T cells. And thereafter they used their one customized construct, that is SCE. In that case, they use the peptide signals of this HLA-G, and at the same time the antigen sequence are from this HLA-E, and they are able to see that this guide RNA 13 will cause a suppressions of this HLA-E expressions, but there is not much effect shown for when they use the guide RNA 1. So indicating that this guide RNA 1 can suppress the, so far the guide RNA, it seems like guide RNA 1 can suppress the HLA ABC expressions, and also this guide RNA 3 can do the same thing, but this guide RNA 1 has a minimum effect on the expressions of this HLA-E, whereas the guide RNA 13 can suppress the HLA-E also. Now they wanted to do some cell killing assays, so they took the allogenic NK92 cells. So they did it at 2 different conditions, or 2 different cases. First one, they took the NK92 cells which is the allogenic NK92 cells, and they isolate the T cells of the patients, and then prime the T cells with the allogenic NK92 cells for 7 days in the presence of IL-2, and then co-culture them and see the effect, whether this T cells is able to the T cells is able to affect this NK92 cell, this NK cells with normal expressions of this HLA ABC and with the suppressions of this HLA ABC. And on the other hand, they also did the NK cells killing assay with NK cells of the host. In that case, they checked the different expressions of this HLA ABC with this NK92 host cells. The results, the panel H results shows that for the normal expressions of this HLA ABC, they are able to see more NK92 killings, whereas when the guide RNAs worked and there is suppression of this HLA ABC, they are able to see minimum killing, around 10% of the around 10% of this NK92 cells gets lysed in the case where guide RNAs are used to suppress HLA expressions. However, from literature, they are able to see that in the NK92 cells, there is a pathway that prevents the NK92 cells killing, and that is like something like SRP alpha pathway. And that SRP alpha pathway, when CD47 and SRP alpha pathway, and this thing they wanted to check its the effect of this host T cells and host NK92 cells in the absence of that pathways. They also want to check that in order to inhibit that path or abrogate that pathways, they use anti-CD47 and anti-FcRA against anti-FcRA to prevent this SRP alpha and CD47's neutralize the SRP alpha and CD47's high expressions on this NK cells. And in that case, what they are able to see, completely different results. That is, in the case where HLA ABCs are expressed in the normal rate, they see minimum cell death, whereas in the case where they have used or they have reduced the expression of this HLA ABC, they are able to see more cell death. The reasons so far they have not explained till the end of the description of this figure 1. We will continue our discussions in our subsequent videos to understand what is the reason behind this opposite behavior, especially on the cells where normal HLA ABCs are expressed. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.